Good afternoon, everyone, and welcome to our second seminar of a Women's Health program that we have at the Media Lab. It's an absolute pleasure to invite our dear friend, and I would say, for me, Linda is one of the professors that I really admire. Her work, her personality, her passion, everything that she does to create awareness on women's health. So it's my victory to have you at the Media Lab too. We worked a lot of time to have you here, and finally we made it possible, and it's a great occasion to share this moment with all of you. Thank you so much for coming. I would like to give a little intro about Linda. She's extremely successful that I don't know where to start, but she has almost everything. She's a member of National Academy of Engineering, National Academy of Medicine, National Academy of Inventors, the American Academy of Arts and Sciences, and she's a MacArthur Fellow. I'm not sure if you know about those fellowships. They are given away to, the genius people, and only a limited number of people in every year, and she's one of those. And she has many awards as well. But what I really appreciate about her work is that she brings a rigor and passion on women's health, and specifically she directs the Center for Gynepathology Research here at MIT, and she has commercialized several tissue engineering platforms, microphysiological system platform technologies, including the 3D printing for bone generation and the liver chip. I'm sure she will touch base on those things. And very recently, for the company that she's a co-founder, Lumicell, which received an FDA clearance in 2024, very recently, for the technology to detect tumor margins intraoperatively. So she will also, I believe, talk about those. So we are so excited to have our dear friend Linda to give this important seminar today and share everything that she has done so far with us. Thank you, Linda, for coming.
So much. Thanks. It's really fun to be here. The most times I've been in this room have been for the McVicker Day events, which is a teaching award, and my thing I'm proudest of is actually being a McVicker Fellow, which is an award given for undergraduate teaching, and we would often have events celebrating each year undergrad teaching at MIT, a very, very important part of research here at this institution. So what I'm going to do today is a little bit of a different talk than I often give, because I want to inspire everyone to be thinking of ways that you can connect your work to gynecology and to certain kinds of problems in women's health that probably emanate from the need of humans to have deep placentation in order to reproduce, because having the placenta go so far into the uterus creates an immunological state in the human women that's very different from men, and creates situations where women are more likely to have chronic inflammatory diseases, adverse responses to vaccines, and certain other things. And these are all things that at MIT we could tackle these problems and really help make progress in preventing problems and diagnosing them and treating when they arise. So there's a lot of great stuff here at the Media Lab that you may not even know could apply. And my job today is to give you an overview of some things we do and ways that we have connected to other people around technology, and also would like to continue doing so in the future. Okay, so I'm going to do just a little bit of stage setting to give you perspective on my background, because I didn't really work in women's health until midway through my career. And I did my host here did a postdoc with Bob Langer. I was his first tissue engineering postdoc, and continued to work with clinical collaborators after I started my faculty position, which was in 1991. And this is a photo of probably the most famous experiment I ever did, the human ear on the back of the mouse. I made the polymer scaffold. So again, like Canan, I went to MRS every year, and this lovely work was first published in an MRS proceedings. But this was a regenerative medicine heyday when it first started, and a lot of my work was in this space, 3D printing for liver regeneration, and we started a bone regeneration company. But I had an epiphany around the time I got tenure that, tissue engineering to replace a liver or so on, would we really want a liver grown in the lab, or would we want to prevent and cure liver disease before you ever would need a transplant? So I turned my attention to building models of human organs, little miniature models we now call microphysiological systems, And starting in the 1990s with regular things like deep reactive ion etching, shown here on the left, our first chip was actually a silicon chip, we developed a way to grow 3D little liver-like tissue in vitro. This shown here is rat liver, and our first reactor, you see those little ports sticking out. It had an external pump. But then so many people were excited about having liver that would keep its function for a long time in culture and that you could circulate medium through. We adapted it to a multi-well plate format that we called the liver chip in 2009 and developed some very, very powerful microfluidic pumps with Dave Trumper in mechanical engineering, and then commercialized it. And it's now something called the physiomimics that most major pharma companies use in order to do preclinical evaluation of drugs that are going to go into humans. And they also are using it more and more for modeling liver disease. We have a big project with Novo Nordisk around cardiometabolic disease that they built a lab in Watertown just to interact with us and give us about $1 million a year to do. But we were able to develop technology that's so widely used for a couple of reasons. The real standard at the time we started working is shown here. And these are chips made at Harvard, of course. They're little cute PDMS-style chips. And around the world, people do tissue engineering in these microfluidic chips, including our own lab. And you're saying, what has Bob Metcalfe got to do with this? Bob Metcalfe won the Turing Award for his contributions to inventing the Ethernet and contributing to the internet. And I have this here because one of the things that we did when we started is we worked with pharma and end users around their needs. And this led us to anticipate the most challenging problems, which included things that ultimately impact women's health. And I get inspiration from Bob because when he was developing the technology in 3Com for commercial use, his engineers wanted it to withstand a lightning strike in the building. And there was a lot of debate at the company because it delayed the rolling out of the technology. But sure enough, a company bought his technology and a competitor's and put it in a building, a huge skyscraper in New York. Lightning did strike. The competitor's technology completely melted down, and the 3Com technology stood up, and that really made him successful. So this is the MIT ethos, to think about what is MIT hard, how do we build around that? So we did not use PDMS because it absorbs anything lipophilic, and over half of small molecule drugs today are lipophilic, so you would not be able to do pharmacokinetic, which is what drug companies really needed at the time. So we did this hard plastic. This model, the liver chip, doesn't look cute, but boy, did it give you fantastic data, and it's also easy to use. So it's withstood the test of time, but I'll show you our newer versions that we're working with Novo to take on board that can do more biology. Okay, while I was doing all of this as an assistant professor, when I first started, I was hired by HST, and they lost my lab space. So I went around, I found some really great space in the old Building 20 near the Biotech Process Engineering Center. And it wasn't quite big enough, but there was this ladies' room with a huge lounge area with couches in it. So I went to the provost and said, can I annex this space? Because it would be so great, then I'd have a perfect lab. It's like, no. You can't take that space because, and this was 1993, women need to lie down every month. That's what the provost's office said. And not because they were mean. They really meant they need to preserve this space because they knew it was a, open secret that a lot of women actually did really need to lie down every month when they had their periods, okay? So keep that in mind. And then fast forward a few years, I got tenure, obviously, and doing well with all the liver chip and so on. And my sister, who lives in Atlanta, started calling me when her daughter started her periods. She's 12 or so. Terrible, terrible problems, bleeding, horrible pain, passing out, throwing up, et cetera. This continues. She gets put on birth control pills. She doesn't get better. And her female OBGYN eventually tells my sister, your daughter's making everything up to get out of going to school. And she was the best student at her school. And so I got her referred to a surgeon in Atlanta, and she had So what was really interesting, I got very hot and bothered by this for a lot of reasons, and they've been kind of percolating. I was not at all working in women's health, but a book published the year before Caitlin was born. She was born in 1990, and 2 years before I started on the faculty, is this. And the book has a section on endometriosis. I taught endocrinology when I first started here at Harvard, because I was an HST, and this was the book that we used as a reference. And let me blow this up for you. And here's what it says. The typical patient with endometriosis will be a nulliparous, meaning not having had kids, in her late 20s or early 30s. Textbooks like this one describe her as intelligent, egocentric, over-anxious, and a perfectionist. Okay? I had been diagnosed with endometriosis a year before this book was published, and it was an accident. I was 28. Okay, so this is what we call a diagnosis bias. Okay? We know now, the cognizati who work in this area know now that the symptoms actually usually start before you even start your period, typically with really bad stomach problems, GI problems. Then you progress to having terrible menstrual problems, and so many young patients are shunted off for, like my niece, for colonoscopies or other bowel things. So I'm on a mission to help raise awareness that this is a pediatric disease. We desperately need better ways to monitor young patients, minimally invasively or non-invasively, to understand what's going on in their abdominal areas. And I've had some lovely discussions over lunch about how we might do this, but if you have any ideas, come see me, because I keep track of all of this. So endometriosis is a disease, and I'll show you more about it, that really does start in the uterus. And so this is the endometrium, or the lining of the uterus. So you see up on the left, the uterus is mainly a muscle. It's got an outer muscle that pushes the baby out and an inner muscle that moves fluid around up or down inside the cavity. And it's a single layer epithelium that's supported by stroma, meaning fibroblasts, blood vessels, and immune cells. And you can see in this beautiful light sheet imaging from a group in Japan, where they imaged the endometrium at the time that you would normally have an embryo implant, called the secretory phase. The yellow are the epithelial cells, and they reconstructed this to show the structure of the glands in the endometrium. And they completely revolutionized how people think about this, because the upper 2/3 is shed and the bottom third stays every month. So what I'm not showing is there's amazing immunology that goes on in this organ. Amazing. I'm happy to talk to people later about that. Now, endometriosis is one of the diseases that happens when the uterus has got some kind of pathology. And it's characterized by the ectopic, or outside the uterus, growth of tissue that resembles the lining of the uterus, or the endometrium. And it's a chronic inflammatory disease. I'm showing a couple of types of lesions. Very vascularized lesions on the abdominal wall, and also lesions that have invaded the muscle in the bowel. And so it can invade through the diaphragm. It has a tropism for stiff tissue. It typically doesn't go all the places that ovarian cancer does. And saying 1 in 10 women, that's prevalent. 10% of women have this, and I'm not being inclusive, because I actually should say and girls, because it is, and I didn't modify this slide. The therapies right now are not so great, or I wouldn't be talking to you. They all focus on suppressing estrogen, high progesterone, menopause, anabolic steroids, et cetera, or surgery. And this is a picture taken one day when I was collecting samples from patients at Newton Wellesley Hospital with the surgeons I collaborate with. Worse yet is that there are other gynecology diseases. Here, this is adenomyosis, and it's a special kind of ectopic endometrium, so it's the sibling of endometriosis, and it's when you get these lesions in the muscle of the uterus itself. And this is a photo from one of my former postdocs. So you can see this beautiful epithelial structure, fluid-filled lumen, polarized epithelia, sitting in muscle. Okay? For those of you who study cancer, carcinoma doesn't even invade muscle like this. So this is a really, really interesting tissue from so many angles: biomechanics, biology, everything. Now, I want to call your attention to some statistics that tell you how horrible it is to work in these areas, because there's only ever been 2 NIH grants on adenomyosis, okay? And if you use PubMed as a metric for aggregating all the research around the world, and compare adenomyosis to a disease with comparable morbidity, disease-adjusted life years would be the metric, you can see there's only about 5% of the research intensity in adenomyosis compared to endo. Most surgeons don't even look for adenomyosis when they evaluate patients for endo. So there's huge, huge, huge diagnostic problems, treatment problems. which means opportunity for any young person in this room, and that's pretty much everybody here younger than me. All right, so how did I get pulled into this? Well, all of this is swirling. My niece, various things at MIT, my own situation, and Susan Whitehead, who's on the corporation, together with Desh Deshpande's wife, invited me to do an event at the Museum of Science, and they said, because I was working on bone tissue engineering and liver, I was doing 3D print, all this cool stuff, no women's health stuff. But at that event, a student asked me, where do you see your lab in 3 to 5 years? And I blurted out, I'm going to have a lab, I'm going to found a center that studies endometriosis. And I didn't know where it came from, but I had just gotten a MacArthur Award, and you're supposed to do something creative, and so it's like mad money. I flew people in, I started talking to people. So in 2009, we got a grant from a foundation, we being my clinical collaborator, Keith Isaacson, shown here. He just recently moved to New Orleans, but he was at Newton Wellesley. And so he called me and said, I just operated on a 23-year-old girl, and it's the 10th surgery she had for endometriosis. And so that was it. He convinced me because he'd been bugging me. It's like, okay. And he also said, animal models don't work. We're stuck in the same old drugs. Nothing is working. We need engineers to come in and work on this. And so we set it up as a way for clinicians. I knew nothing about this disease other than my own personal experience, but for a way for clinicians, scientists, engineers, everybody to come together and share disease and chart new paths. Okay? And we were joined by the first department head for biological engineering, Doug Laufenberger, who was pioneering methods in systems biology. I'll get more to that later. It was very important because I'm not the kind of engineer, like a lot of you here are, that builds devices. I think about biology. And we do biomaterials, but that's a lot of molecular interactions with cells. And fortunately, MIT is an incredibly special place to do this kind of work and have this kind of center because we have the infinite corridor effect. And it's a little bit harder today because there's a lot of more spread out MIT, but we definitely still have a lot of ways to interact with colleagues at various events, including things like this. But there's also a huge emphasis on undergraduate teaching here and teaching across department lines. So I led development of the undergraduate major in Course 20. We had to do a lot of teaching partnerships because we didn't have a lot of faculty. And I had the great pleasure of teaching with Munji Bawendi, who some of you may know from inventing quantum dots. He won the Nobel Prize recently. But we taught physical chemistry together. And in the course of thinking about fun exam problems and homework problems, we also thought about research. So together with David Lee from the Koch Institute, we came up with an idea of how to image residual tumors when you have a lumpectomy, particularly for breast cancer. So if the surgeon wants to do breast conserving surgery, there's a concern that maybe she doesn't get all the tumor out. How do you find that? And so one of the most stressful times for a patient is waiting to see if your margins were clear. And I've been through breast cancer twice, so I know this nervousness. So we founded a company that lets you inject the patient with an imaging agent that gets activated by the tumor cells so that you can scan the cavity when you take the tumor out and decide, is there still tumor there? And keep shaving stuff off until it's all clear. It's better than standard pathology, and I really regret that the clinical trial had just finished when I had my last lumpectomy and the FDA was still pending approval. But it's fantastic. Anybody you know who has breast cancer should see if they can get this. So the other special thing about MIT, compared to other institutions for this particular kind of endeavor, i.e. connecting with clinicians, is that we started a new discipline of engineering called biological engineering. And I want to give you a little sense of this because it helps you orient to who's doing what on campus and how to just talk to them. So when I was a student, there was no much bioengineering. I majored in chemical engineering, but there was biomedical engineering, and it's really medical engineering where you have all kinds of engineering disciplines applied to problems in medicine. You don't really need to know biology, but you can do fabulous things. You can do the kinds of things that are done here in the Media Lab with ultrasound. All of these fabulous things that will help patients can be done interacting with patients through application of mechanical engineering, mechatronics, electrical engineering, and so on. and the substrate sciences are generally physics, chemistry, and math. When we started biological engineering, it was really around the idea that biology had become molecular, and you needed a way to better organize all of the information flow in biology. All of these multi-omics also intersect with biophysics, biochemistry, things changing in space and time. So it's not just AI, you really have to have a grasp of phenomena and the physics of the phenomena together with the biochemistry and not just data. So the ability to look at biology and translate biology through an engineering lens of analysis, design, and synthesis for application not only in medicine but agriculture, materials, et cetera, is what drove creation of biological engineering in a very organic way here at MIT, because we had to create curricula to do this. And so now biology is a substrate for biomedical engineering, and sometimes the word bioengineering is used to have all of these kinds of things interchangeable with biomedical engineering. And just to finish up this bit, this has actually been recognized by the National Academy of Engineering as a new discipline of bioengineering, of engineering. So again, biomedical engineering, you don't really need to know particular underlying biology. You probably need physiology. In biological engineering, you need to know things about how cells are processing information. You need to be able to model what the cell is doing in some kind of mathematical way. It may be a data-informed way, or it may be a mechanistic, deterministic way, but some kind of mathematical models. And in 2021, the National Academy of Engineering gave MIT an award for creating, the highest award for education, the Gordon Prize for creating a new discipline of biological engineering. So we should be really proud because other schools copy what MIT did. All right, now I want to give a little example because I want to try to tie this to the Media Lab, and particularly I'll end up on Roz Picard. But we have to do a little weaving to get there. And so let me just give you an example. There was a blockbuster Amgen drug, Neupogen. It's a molecule called GCSF that creates white cell blood growth. So if you have cancer and you're going to have dose-dense ACT, big chemotherapy like I did when I had my first breast cancer, you have to have this drug to stimulate your white cells to grow or you'll get a fatal infection. And so this completely transformed the kind of chemotherapy that people could do. But around the early 2000s, late 1990s, 2000s, it was going off patent, and also patients had to take it every day. It was really hard to administer. So they wanted to redesign this drug to have better pharmacokinetics. How can we give it so it lasts longer, patients don't have to come in all the time? And so there was a big project with MIT and that Doug Laufenberger led and that Bruce Tidor was part of. How do you redesign GCSF, one, so you can patent it, but also so that it has better pharmacokinetics as a drug? And they ultimately came up with a drug called Neulasta, and that's actually the drug I took. And I teach this redesign in Thermo, so let me just give you a little bit of picture so you understand a little bit about biological engineering, and that we'll connect it back to Roz at the end. So you use something called quantitative systems pharmacology. This is a field of, you could call it pharmacokinetics, but it really brings in biological effects, typically of biological agents like Neulasta, which is a protein growth factor. And so here, you need to think about the drug across multiple dimensions. And I'm not going to go into it all here, but there is a way that we organize the information flow. So it's not just AI, it's thinking about things that the cell is doing dynamically to the drug and that the drug is doing to the cell. So you have to know some biology, and there is some deterministic modeling that will come in. So you can combine AI kind of stuff, but with deterministic. So QSP almost always operates partly in a deterministic, mechanistic state to understand how to improve something and actually how to apply it to patients. So you got to move beyond AI and incorporate more of the physics of space and time in this. So the real breakthrough came with years of work that Doug had done on how a drug like GCSF binds to a receptor on the surface of the cell, signals, and moves through the cell to either be degraded or recycled. And Amgen and other companies often thought you want the drug to bind tighter. It turned out that was not what you wanted at all. What you really wanted was for the drug to recycle, okay? Because most drugs, they bound tighter, they would stay bound, and then they would get degraded. And Doug found out if you allow them to fall off inside the cell, they get recycled. So they used that criterion, and they redesigned G-CSF so that it would fall off inside the cell. So it would still bind the same tightness at the surface, but it would fall off inside due to a pH switch. And they did a lot of modeling, and they got a patent on this, and they showed that it would work to improve the PK, the pharmacodynamics, and improve in mice people. Now, we're going to come back on slide 42 to a connection back to the Media Lab, okay? And I'm going to go through these slides. Don't worry, I'm going to go through these. So what did we do in terms of endometriosis? And working with Keith Isaacson, it was scary. I really knew nothing about the biology. We linked up. I went on the NIH Reporter, found people who had grants, asked one of them would they advise us. He said yes. So they helped us think about inflammation in the peritoneal cavity. So you can see on the upper left, this is a patient having surgery. There's blood in her peritoneal fluid. There's always some fluid in the abdomen. You can see her lesions there. We studied this in about 100 women, about 20 controls and the rest, patients who most of them were not taking any kind of hormones, but some were. And we did machine learning on 50 of the immune molecules in the peritoneal fluid to identify a subgroup, about a third of the patients, who had a signature. So it was a multivariate, unsupervised analysis to get the patient groups. Then we found a signature that we could use bioinformatics to discern that a particular kind of cell in the abdominal cavity, shown on the lower left, a macrophage, was making these inflammatory molecules that was causing this pain and so on. And moreover, there was a particular enzyme inside of the cell called Jun kinase that was controlling this. So this was very, very exciting for us because controlling inflammation, instead of giving the patient all these crazy hormones, might be a better route to actually fixing the symptoms with fewer side effects. And then we did some other clinical studies to implicate it in invasion, and we were shocked to learn that after we published this, that in fact a Jun kinase inhibitor had already been used to cure patient populations, and here they are. The Steve Palmer here is a very, very interesting scientist. At the time he was at Merck. Merck was developing these inhibitors for other inflammatory diseases, and Steve Palmer got them to do these two preclinical models of endometriosis, and it worked great in the rodents and baboons, but when they licensed it to a company called Preglim, and the clinical trial was not successful. And so there were maybe some, the drug could be a little bit better, but the clinical trial design is also very difficult. It wasn't a great design, and so it just didn't meet the clinical endpoints. But there were trends that made it look like a very promising target. So I was giving a talk at the American Society for Reproductive Medicine meeting in Hawaii in 2014. There was a hurricane, 12 inches of rain a day for a few days. And so Steve and I ended up together in a bar talking about what's next for these Jun kinase inhibitors. And so he's like, I'm going to go to academia. I'm going to make new molecules that I think will be better. What you need to do is go and make an actual model of the endometriosis lesions in patients with little blood vessels, immune cells recruiting. That's what we really need to get this into the clinic, because there's a real concern now that the animal models are not representing humans. What a shock. So I'm like, okay, we'll do that. So what we did, and this is a little of the science, around that time, well, in 2017, but let's just lead the years, we were doing a lot of in vitro tissue engineering, building biomaterials to get 3D tissues to grow. And during this time, there was an amazing advance in the ability to grow the epithelial cells that line the gut and the endometrium and others. You could make tissue banks. When we first started this kind of work, right after I talked to Steve Palmer, when you got a biopsy from the clinic of a patient's endometrium, you had to use the cells right away. You couldn't expand them for a tissue bank. But this technology where you would take the glands, put them in a special mix called Matrigel, and grow them up was a complete game changer, because now we could make a whole tissue bank filled with organoids that have been expanded in culture. But the reagent that people use to support these organoids is really terrible for all the reasons I have listed here. And so people love it because it works, but they hate it because it's got all these problems. So we spent a long time to develop a completely, utterly synthetic, meaning completely made in the lab, not like Matrigel extracted from a mouse tumor, a synthetic hydrogel that would support human mucosal barrier organoid growth. And this was a real accomplishment. So it's just a branch polymer that we have little peptides that interact with cell surface, cross-linked with peptides that the cells can remodel. You mix it all together and you get this beautiful gel that looks like this. And it's really reproducible, it's really easy to use, but it took us 10 years because it's really an engineering problem. There's many parameters, there's matrix stiffness, there's permeability, there's ligand spacing, many, many things that you have to have an appreciation for to do this. This is in commercialization now, and the beta testers have been lined up, and so it hopefully will be in a catalog later this year because people around the world are using it with our help. We publish the instructions, but we also send it to people, and it works, the only one for mucosal barriers. And this is just a couple of fun movies of organoids. These happen to be gut, and I don't have the endometrium. It's a nice thing, but this is in that matrix that people use that's from the mouse tumor, and this is in our synthetic matrix. And there's a lot of really interesting biomechanics about why they're pulsing and what this means for how endometriosis may be growing in a muscle that later I can talk to anyone who's really interested. So what we also strove to do is that most people had focused on just growing the organoid, just that epithelial cell barrier. But in fact, the endometrium is a tissue that all the cells are talking to each other. There's enormous swings in the hormones that they respond to individually, and crosstalk allows the tissue to grow and to be shed. So you really need the crosstalk between the epithelial cells and the other cell types to capture the pathologies and the normal function. So what was really important about our matrix, it was a one-size-fits-all in that we built it so it could support the epithelial cells, but also all these other cell types. And that's extremely important because we ultimately, remember, we need to grow a whole microfluidic lesion model. So I had an amazing postdoc, Juan Necco, who took the matrix and figured out how to grow. These are the epithelial organoids. Actin labels the apical side, the top, and then the red are the fibroblasts. And so in this paper, it was only the organoids and fibroblasts. But they have a completely normal response to the hormone changes during the menstrual cycle. And what's really great also about having a completely synthetic matrix, we can do some engineering of that environment that you can't do with a natural protein. And in particular, we're very interested in how the stiffness of the extracellular environment may affect the properties of the cells. So this is an adenomyosis lesion in a patient. This is her endometrium, and this is the lesion. And if we do light sheet imaging, which is a thick piece of tissue about 1/2 centimeter, and you see those white, that's the epithelial cells in the muscle. They're invading, okay? And that dark area in the middle is fluid. It's just a fluid-filled lumen. With our synthetic matrix, we can make a soft environment like the endometrium on the left, and these are co-cultures, so you can see the organoids look normal. But over on the right, we made the matrix have a mechanical property similar to the muscle, and you can see now that the organoids are taking on an invasive phenotype. Now, I carved my Halloween pumpkin with the following, inferring causation from correlation. So we're not going to say everything is due to stiffness. We're making orthogonal materials to test the mechanics, but there are some things that hold up about this. The key thing to lesions, though, is they're very highly vascularized, and you saw that in some of the lesions I showed you before. There's a light sheet image that Juan Necco did when he was a postdoc showing one of these lesions completely surrounded by blood vessels, which are red. The green, we need to come in and build blood vessels to support the lesions. It's not enough just to have the organoids and fibroblasts. We need these lesions. So we leverage some really pioneering work by our colleague Roger Cam in biological and mechanical engineering, who over decades had invented a way to take a microfluidic device, little plastic chip, where there's a channel on either side, and in the central region, he figured out how you could inject a liquid that would turn into a fibrin gel, so it would have fibrogen and thrombin in it, but with vessel cells, endothelial cells, and fibroblasts, such that this liquid was held in place by surface tension until it formed a gel, and then over a period of about 4 days, the vascular cells would organize and form perfusible vessels. And what you're seeing here in this movie, which unfortunately is not looping, was immune cells traversing from one side to the other. And Ellen Kahn is the first graduate student that Roger and I had joint together, her first experiment replicating things that Roger's lab had been doing for many, many years, and it's just here to show you. Now, a limitation of this is it's fibrin, and also it doesn't have pumps. This is all gravity flow. But Ellen developed a model, and she's defending her thesis soon, and she's won multiple awards at conferences for this, of how a lesion can attract blood vessels when it's there. So she's got a beautiful story on how patients with different properties, the blood vessels grow in. And so I'm just showing you this. But she had to do a kludge, grow the blood vessels, and then come back and put the lesion, had to be in the synthetic gel. So it's very clever what she did, but our real dream was to be able to put everything in together and have the lesions there from the start and the blood vessels grow around them in the completely synthetic gel, because it's really important, going back to Bob Metcalf, it has to be reproducible, it has to be easy to use, and fibrin is not that easy to use. So this is a time-lapse video of Lauren Pruitt with others in the lab in a completely nice plastic device in a completely synthetic gel. The organoids are there in the green, and the blood vessels in a synthetic gel are growing around them. And this particular model, as I'll show in a moment, has pumps and everything. So we've transitioned to really what to me is like wild. So coming back full circle, Steve Palmer did go to Baylor. He did create a whole suite of new drugs. He licensed them to Cellmatics. And this is us launching a project together to test his new Jun kinase inhibitors in this model of endometriosis. And it's hopeful that we will be giving them data that will help them advance to address a whole host of problems that women have. We designed our devices. Remember, at the beginning, we can't use PDMS. We have to use hard plastics. So we worked very hard with Dave Trumper to adapt the liver chip pumps to a microfluidic format. There's air and vacuum drive them. And so we've applied this same approach to a whole host of other diseases, particularly to liver. So we grow vascularized liver. We're studying Lyme disease. Chronic infection plagues women more than men. We've got a model of Lyme disease, and these are the bacteria that cause it. We've grown gut microbiome with human colon. So our vision is that for MIT, we would build a core facility run by the Division of Comparative Medicine. So instead of using a mouse, you could use a human equivalent. And that's a vision, and I think we're going to realize it. And a reason I think we're going to realize it is that everybody is trying to move away from mice. They're just not that informative for so many things. So pharma's completely on board with this. This is Lyda Knudsen, who won the Lasker Prize from the GLP-1. She's a friend of mine because we work closely with Novo. And they really built a lab here to interact with us because they know that they need to get away from the mice. Moreover, NIH did a big study, and I was part of this, as was Kelly Pate, who runs DCM, on how do we catalyze the use of alternative methods to animals. And so NIH is all on board with this. They just have finished up a call for proposals for centers that would form a consortium. How do we move these into regulatory? So we submitted a center grant proposal that would be $2 million a year for 5 years to bring these endometriosis drug testing into the regulatory process because they'll have FDA. And the FDA, a couple of weeks ago, this was the headline in the Saturday Boston Globe business section. They're going to phase out animals. Okay, so there's huge pressure. MIT should lead this. We need all kind of technologies for this. So I want to end up around the work that we're doing under the auspices of the HEALS program, and a lot of you may have heard of this, but our project was incubating long before the HEALS was even there because we had a donor who was very interested in these chronic problems that women had and the links between gynecology and things like chronic Lyme. And so our initiative, it really expands the goals of the Center for Gynepathology Research, which is to help engage the MIT community with great problems. I know a lot of clinicians. I know the industry. And I know what a lot of the gaps are. And there's got to be technology at MIT that some of you are working on that could be applied to these kinds of questions. So we have a lot of events. We just had our launch, and we want to build it. What we're trying to do more broadly, and a huge vision, is to do for menstruation and gynecology what was done for breast cancer in the last century. And there were a lot of patient advocates who's, you used to not be able to talk about breast cancer at all when I was young, okay? And then patient advocates together with some congresswomen got together and started really pushing for things. But it was when Susan Love, a surgeon, wrote this amazing book, which is now in its 7th edition, intended not only for lay people but for doctors, about what to expect when you went to be evaluated for breast cancer, that it really put a lot of extra funding and so on to NIH and really catapulted breast cancer into an area of science. So that my dean, Anantha, who's kind of he can say mammary and breast, and nobody thinks tit or boob. But for menstruation, there's still a taboo. So we want to do away with that by bringing great technology in. And so broadly, menstruation underpins this whole problem that we have as humans, in that immunology is very sex and species specific. So we can't really use animals to study. Certainly mice don't even menstruate unless you really go to great lengths. We really need to build these human models like I described. So our vision is that we combine really great clinical phenotyping at the physiological level and at the molecular level with this kind of modeling I described, and then cycle through these living patient avatars until we can come up with mechanistic understanding of the disease and drugs that will work effectively in very specific patient groups. So let me just end with a couple of organic connections that have come about because of our efforts to engage people in the community. And again, we need all hands on deck. I'm not proud. If somebody has something better than what I'm doing in lab, I want to hear about it because I don't want to be working on a second-rate thing. So one thing we do is, for years we've had a gynecology surgery conference. It was started by surgeons. They wanted to come to MIT and do blue sky thinking about their field and share stuff with scientists. And the surgeons would invite all these famous surgeons from around the world, but they really, really loved that I would have people from MIT come give talks, and they were TED-like talks, and they would come do lab tours and stuff like that. And so one of the talks early on was Dina Katabi. Some of you may know her. She invented something called the Emerald system that uses analysis of changes in electromagnetic waves when you move to look at people walking, sleeping, breathing, et cetera. It's fascinating, fascinating. We did a little, she came and spoke at OEF, and then we did a little project looking at how endometriosis patients might have their sleep disrupted. It was very early stages. The Emerald is now a lot better developed, and so we'll come back to this. But this idea of non-invasive measurements of patient physiology is one that we are in such desperate need of. So now I want to come back. This is slide 42. And remember, I told you slide 42 is going to connect to the Media Lab. It's going to take this step first, and then I'll come to the next slide. Okay, so what was really fun for us, in our first paper, where we identified the molecular classification, we showed that a molecule called IL-8, which is an inflammatory molecule in the peritoneal fluid, it was the most important inflammatory molecule in that paper. Okay, so Chugai, which is one of those companies that's kind of owned by Roche, started doing work on IL-8, and they published this paper a couple of years ago where they developed an antibody against IL-8, and they did a special trick. They put something on it that allowed it to recycle. And you know what they put on it? They put the histidine switch that Doug Laufenberger developed. So this was very cute because it related to something I was senior author on and something he was senior. Doug's my husband, so it was really kind of cute, and they came and talked to us. Okay, so they showed that this was better. It's in clinical trials. But remember I said how hard it is to get patient data, patient-reported outcome tools? You really need to get metrics. So what are they using? Guess who gave a talk at OEF? Roz Picard. She gave an amazing talk. The work is amazing, and of course she has a company, Empatica, and guess who Chugai is working with to evaluate the patient symptoms for their FDA clinical trial of the anti-IL-8. Okay, so now if that wasn't a cool way to connect the biology to the Media Lab, okay, so this is incredibly important to get all of these things linked together. And I'll finish up with just what we're doing now, a couple of examples. We through a variety of things on campus, we need to get more info out of our in vitro models. They're very resource intensive to build, and we linked up with somebody in RLE, Sishan Yu, and she is developing label-free imaging. So it's multiphoton, and so there's no stains here. The left, this is a mammary tumor in a mouse. The green is second harmonics for collagen. You can see the blood going through, et cetera. And so we gave her just a biopsy of endometrium, and her student, who knew hardly any biology, started counting the nucleoli. And the nucleoli are the organelles inside the nucleus that make ribosomes. Okay? And he's counting them, and he had this whole thing, and he comes to us, and I'm like, it's a nucleolus. But it turns out they're very important. It turns out the endometrium has a bizarre nucleolar physiology, and it's so bizarre, there's somebody at MIT in biology who studies it, and his name is Eleazar Kalo, and he's associate professor. So we did a hop, skip, and a jump from just a random, it happens only in a tiny window after the progesterone surge. It may be related to the biology of implantation. So he studies this, but he'd been not having real samples. So now we got funding to work with him very much on this biology, and brought in Whitney Henry, another biology assistant professor, because this also may be related to heavy menstrual bleeding. So one little pilot experiment, we now have gotten, I got about $1.5 million in funding for Shishan's lab to build a microscope for us to use here. So this is a way that our center, we do pilot studies if you have skin in the game, and we help you get connected to the problem. And this is really, really fascinating biology now. So this is Eleazar's imaging of these structures. So we also have a team, sex and immunity and infection, and it's around, this is Sishan's imaging of a microbe that causes Lyme, Borrelia, in the collagen of a mouse. So these microbes get into the fascia and hide. You can see it there, and this is, as you can see with label-free imaging. So we're building in vitro models to do this in a human state, because the mouse immunology doesn't interact with the microbe the same way. So we have a fabulous team, again, to study this biology, applying our tools. Preaching to the choir that gynecology is very underfunded. This is a study that was published by Mirren showing that on average, if you plot funding per year versus disease-adjusted life years, the gynecology diseases are much less well-funded. So you had, as your first speaker, Megan Greenfield, who contributed this. I was an advisor on this. I'm acknowledged in both the first and second reports. We're very close to each other, trying to help destigmatize this. So I'll end with a few comments I have on the landscape. There's a lot of interest in femtech. We need to push femtech. It's kind of squishy. I think it's mostly devices and apps. There's huge visibility for women's health. And I think a lot of VCs in pharma, though, are really timid. Chronic inflammatory and autoimmune, and they're really hard to study in animals. And it's just like we've got to get traction on this. And this is why I'm so keen to build this core facility. And we obviously need the kind of biological engineering we do to integrate. I love devices, but I have to do biology. So that people have something to work on. The final thing I will say before the acknowledgments is when I started a center at MIT, it was not well received. It was hard. It was hard because there was no NIH funding, and it was hard because prominent women at MIT came to me when I started raising awareness about the incredible prevalence of these diseases. I mean, 1 in 3 women over the age of 60 in this country have had a hysterectomy, and you don't get a hysterectomy just because you feel like it. You suffer for years before you get one. Over half of women have really bad gynecology problems, and a quarter miss school or work at least 1 day a week. So to this woman here, there were women walking around in Harvard Yard after the first Trump administration when there were those protests, I stopped and I said, congratulations on your excellent menstrual health, but, not everybody has this. And so I coined, I call this period privilege. I think there are people who don't appreciate how hard it is for a lot of women, including women in the MIT community, because I hear from a lot of them. I connect them to the help they need. So appreciating how prevalent these problems are and how much people suffer, usually silently and even being shunned, is really important, and we can change that. I've only recently at MIT, my center was used as bait to get donors to come fund the Koch Institute. So it's only recently that I've been allowed to raise any money. So this is the end. We will change biomedical research if we focus on women's health. We're having a moonshot for menstruation science. It should actually be a Mars shot because Elon Musk wants to populate Mars. The women need to be 100% fertile, so we need to solve all these gynecology problems. I actually tweeted that, and I did get funding from his foundation. So these are my acknowledgments. I'm very grateful to Emily Fairbairn, who is a citizen scientist. Her husband's an alum of MIT. Their whole family had chronic Lyme. She's an entrepreneur. She and her husband have a donor advised fund that funds this new initiative that we have. She's a real person of hope, and she's fantastic. We interact with her all the time. And then I have my team of people, including Doug Laufenberger here with his first student who studied endometriosis, and Dave Trumper, my mechatronics collaborator, and then my clinical collaborators and people around the world we continue to work with. Thank you. Or I can talk to you later if you're, this was an overview. Yeah. Yes. So molecular classification of disease. Yeah, okay, so great question, because I didn't really say anything about that. So when we did that study, we started out, I actually got breast cancer right after we started our center. I got breast cancer, and I was triple negative. And I found a tumor myself in Singapore. Now, if I'd had a conformable device, I probably would have found it earlier. But within a week, I knew I was triple negative. I had my surgery plan, and I had my oncology plan. I knew. And when you have endometriosis, it can take years to be even taken seriously. So we got the idea that because 10% of women have it, there must be molecular classification after I got classified as triple negative. And so what we went in is like, let's study the peritoneal fluid, and there must be different immune networks in different groups of women. Now, we only had about 100 women, which is a very small study. For cancer, you'd have many, many more patients. And so we only got one group separate from another group. None of the controls showed up in our signature. So that first group was around immune signatures. Now, I'm not saying it's the best way to classify patients, but we did replicate it in a study in Brazil. But what it did is it opened up, as often in science, we published it. It was very hard to get published. We did publish it in Science Translational Medicine ultimately. But it got people thinking that there must be molecular subgroups, just like there are for Crohn's and all these other chronic inflammatory diseases. You know, with Crohn's, if anybody knows somebody with Crohn's, some people respond to Humira. Some people need Skyrizi. You know, there's all these different pathways. Some of them may be related to genetics, and so more and more and more GWAS studies are coming out, or genetic linkage studies with variants in SNPs that are associated with disease. And there's some fascinating things, and I work very closely with the geneticists. So the endometriosis community is very tight. So I work very closely with the top people in genetics. And so there are variants in but what we do know, and I didn't mention, is that the way clinicians do it now is that's what we do with endometriosis. If you're stage 4, it's because you have an endometrioma and a lot of other stuff. Okay, and when we submitted this Science Translational Medicine paper and the reviews came back like ready to kill us because we wanted to change a staging system, the surgeon we work with said, you know, it's like, I asked, well, what does the staging really mean? He goes, it's a way for surgeons to talk to each other about how bad it was in there. That's really what it is. And it's not really related to symptoms or severity, because a stage 4 patient may have no symptoms, and she may get pregnant, but a stage 1 patient may be completely crippled in pain. And so we're trying to study that. And so what we need are better molecular classifications. It's probably not going to come from the blood, but we do need the classifications so that we can gate patients to disease. So what I'm doing with companies now, and what we just submitted this big NIH grant for, is to take very well molecularly and pain. Pain is a huge, huge thing that you can phenotype clinically. There's a great group at Michigan. I can talk about pain all day. But to take the pain phenotypes, molecular phenotypes, and say, okay, we think may have, you know, the IL-1, and it's not really IL-1, but there may be different molecular subtypes that would respond to some tweaking of a particular part of the immune system. We're in early stages, but I think it will happen in the next 3 to 5 years. And then to get the drug to match the patient, you will probably go through an era in regulatory where you use models like ours to test, do patients with this particular constellation of molecular signatures respond to this drug? So that's where we are right now, trying to match those. And then you would just have some kind of test that the patient would have at the time. Then you would say, okay, we can test for this immune signature in all patients and tell them which drug to take. That would be, but that's probably 5 years down the road. But I think it will happen. Mm-hmm. Mm-hmm. It's not commercially available yet, and so they're still setting the pricing. It will probably be comparable to Matrigel, though, I can tell you that, because you have to do a lot of quality control. I would, I'm coming from an orthogonal perspective, which is that I'm working on problems that right now there are no solutions for people with these diseases. People suffer. They die, literally. And we have a pharma project, I can't name the company, but the scientist there has championed it because someone close to him died from the complications of endometriosis. It's very sad. So there's applications where being as expensive as Matrigel is not a problem because it's replacing something that doesn't give you information. Right now, what the FDA, what the NIH want, they want reproducible, easy to use, and high information content. So Matrigel clouds information going back and forth between cells. And we have a paper in preparation testing a drug for heavy menstrual bleeding that's not approved in the US, but approved in the UK. And we compare Matrigel to our system against patient samples. And our system is so much better at capturing what actually happens in response to hormones and the drug because we don't have interfering TGF-beta and blah, blah, blah. Okay, so I think my view on going into maker spaces, I don't think about it a lot because I want it to be cheap and turnkey for drug companies to use. So I want Novo, I want whoever, I want them to have the confidence. We have to show the confidence that stuff actually works. So I'm in a very focused area of I want these to be cheap and accessible because I want people to use them to replace animals. And they're only going to do it if it's, cost effective. And companies are starting to do those kind of calculations for select applications, and they're using the in vitro models now instead of animals for certain things. And it's really gaining steam. So when it'll end up in the makerspace, and I'm an advisor to our biomakerspace. I love our biomakerspace. I use it for teaching. But I think it's going to be a while before we have, because a lot of it is problem definition. What is your use case? You need to make something for the information that you need. So we go in and ask, is there an easier way to do this? We always ask, is there a simpler thing than this very complicated microfluidic device? And if there is, we're not going to do the microfluidic device as cute as it looks. Mm-hmm. So I would probably not, I'm not the one to do that. So what I believe firmly in is something called staying in your lane and keeping track of what's going on in the other lanes because, you just never know. But there are people, so Miki Tall, for example, who's part of our team, she's an immunologist. She got her PhD with Akiko Iwasaki at Yale, who's very famous for all the COVID stuff, and then she worked with her Weissman. But there's a whole community of people. Now, what I can contribute are tools to help us do a better job of human immunology and doing sex-specific differences. And a huge problem, believe it or not, is getting steroid hormones to be properly controlled in culture. So we're spending $2 million on that over the next 2 years just to do a baseline project so we can really, really control the hormones properly, because nobody's doing it right. But I would say as a baseline, there's a fascinating guy, Petter, what is his last name? I just went to a beautiful conference at the Sanger Institute in the UK, where there's a lot of interest in this at the molecular and cellular level, where they're studying transgender individuals who have XY chromosome but take female hormones, or XX chromosomes and take male hormones. And what's coming out of that is really interesting, because it shows that, I want to say, the women, I want to say, I might get it backwards, but TNF alpha dominated, and the men are interferon gamma dominated pathways. And it has to do with a particular plasmoid, what is the cell? It was January that I saw this, and I had the flu when I was over there. So I don't remember exactly, but I can say that there's starting to be a lot more insights into what's going on. But the upshot is that women tend to get more chronic inflammatory diseases because their innate immune system gets more activated by things like vaccines and so on. So then the adaptive immune system is running around looking for something to attack, and it builds up responses to self molecules that look sort of like the target bacterium or whatever, but are really the human, because all the antibodies and so on have low affinity for many, many different antigens. So I'm happy to talk to people about immunology. I know enough to give a basic lesson, but not at the detail that Mickey or somebody would.